Things seem to be back to normal. You'll find out later what's green and glows. Okay, if you remember, we've already covered 3 of the 4 steps in genetics research. Step number 1 is identifying disease-causing genes. Step number 2, producing animals with the defective gene. And step number 3, developing and testing new treatments like gene therapy in animals to make sure they're safe and effective. Before we take a look at the last step in genetics research, we'll take a look at another new treatment, stem cell therapy. Imagine being able to repair a damaged heart, grow replacement skin tissue for burn victims, replace the defective cells in the pancreas of a person with diabetes, or replenish the blood cells of cancer patients after they've had chemotherapy. What makes these medical breakthroughs possible? Stem cells, like these. Stem cells are blank cells, which can become other types of specialized cells, and can copy themselves for long periods of time. Many people are confused about what size embryonic stem cells are. These are microscopic images that are magnified so that you can easily see them. So, to give you an idea of the actual size of an embryonic cell, the stage at which the embryo is in when it is used for research is no bigger than a grain of sand. These little cells can play a big role in the medicine of the future. Maybe you've heard about it. Stem cell research is in the news a lot these days.
Really, I think the media plays 2 functions. First of all is the obvious one. We sort of inform the public to some extent of what is happening, what is interesting, what are the trends in the field, and we do it in a language that's not the complicated one used by scientists, but one that's more reader-friendly. Secondly, for the scientists, what we do for them is sort of get their name more in the public's mind and in politicians' mind, and that helps them in the sense that it helps them get more research funding.
There are 2 main different types of stem cells, embryonic stem cells and adult stem cells. Embryonic stem cells, they're derived from fertilized eggs, and they have a very vast potential to change into any cell type in the body. Therefore, they have a huge possible impact in the future of medicine. These cells also have the ability to not change and maintain themselves in an undifferentiated form for very long periods, almost indefinitely. Therefore, they could supply an unlimited supply of cells for regenerative medicine. The second type of stem cells is the adult stem cells, and there are 2 different types of that. One is derived from the bone marrow, and the second from the placenta or umbilical cord. These adult stem cells, they have more limited potential to change into different cell types in the body. So they may not be able to change into bone cells or brain cells or all the different heart cells that embryonic stem cells could change into. So therefore, they have a more limited potential, but within a more defined area, they could possibly also have a major impact in medicine.
A lot of times with things that become controversial like cloning or stem cells, there's not a basic understanding of the science. There's a lot of rhetoric, but maybe there's not a great understanding of what scientists generally want to do and what they need to do that. And so I think a science writer plays a pretty important role in communicating that to the public.
When I read about stem cell research and heard some discussions about it, I started to realize maybe I really didn't understand where researchers can get stem cells.
Where do stem cells come from? First, they're embryonic stem cells, which come from fertilized eggs that have undergone a number of cell divisions. These cause the most discussion, which we'll talk about in detail later. These fertilized eggs are extra eggs that were created for in vitro fertilization. In vitro means in an artificial environment, like a laboratory dish or test tube. The second source of stem cells comes from adult tissues, with the most common source coming from bone marrow, the soft, spongy material in the cavity of the bone. Third, stem cells can come from the umbilical cord and placenta.
The shortcomings of embryonic stem cell research is that there are some federal regulations that limit their use. In particular, the derivation of new stem cells is not allowed. And also there are a lot of ethical issues with regarding destroying life. Adult stem cells have a great potential in that case because they do not have the ethical issues and other problems associated. Adult stem cells also have an added advantage in that immune rejection by the recipient may not be a problem as they might be in embryonic stem cells. Embryonic stem cells, just like tissue donors and acceptors, they might have immune rejection when they're used in therapies. But adult stem cells that would be derived from the person himself who would be needing the therapy would not encounter this immune rejection problem.
Yes, it's time for the answer to our trivia question that you've all been waiting for. What is green and glows in a genetics research lab? The answer is genetically altered stem cells. Because it's important to understand what happens to stem cells after they are injected into an animal, scientists sometimes place a gene into the mouse stem cells that makes them glow green. Although there are other methods, color is the most advanced and the best visual that makes it possible for scientists to track where the stem cells go and how they repair damaged tissue. There are several problems here that researchers are trying to solve, and what they mean for the future of stem cell research.
There are several things we need to look at as we continue to carry out stem cell research. First, we need to learn more about how to change stem cells into specific cell types. And then we need to figure out ways to produce large quantities of these cells in the lab that are uncontaminated and are pure cell types. Another research question we need to address is figuring out ways to modify embryonic stem cells so that they won't be rejected by the immune system.
What begins in a little mouse can have a big impact in the field of medicine. For people like George, it could mean the promise of a cure for diabetes in the future.
I've been hearing about the stem cell research, and it looks really positive. If they work everything out and research keeps going, it could really make a difference. It takes years of testing and approval, but I like to think I'll see some changes in my lifetime.
Our search for the link between genes and diseases now brings us to the last step in the research process: human clinical studies and disease prevention. What does this mean? When animal research has identified a cure that seems to work and is safe, it can be tested on humans following well-established guidelines. It is hoped that some of the animal research you've heard about in this program could eventually be applied to humans. This is the ultimate goal of lab research. Genetic information from research could also be used to help people prevent certain diseases in the future. Imagine, for example, that research on animals shows us that the life-threatening part of heart disease happens just when an animal with the genes for heart disease eats high-fat foods. This type of information can be applied to humans as well. If people have the genes for heart disease, their physician can recommend that they start and stay on a low-fat diet. This kind of knowledge could save lives.
Research in genetics constantly leads to finding new treatments and curing diseases. Even more could change by the time I start a career.
The steps in genetics research are part of a cycle, from identifying the genes that cause diseases, to developing treatments in animals, testing these treatments in humans, and finally coming full circle to screening the population for genes which cause disease, and then preventing diseases with new treatments and cures.
Today we're going to talk about ethics and policy in genetics research. First, Rolando here will tell you the top 10 advances in biotechnology.
You mean me?
Yes, you. Who else?
Well, how about him for starters? Or him. Or him. Or him.
So that's a pretty bizarre nightmare I had last night, huh?
Nightmare?
Well, think about it. An exact copy of yourself, wouldn't that be totally off the wall?
Well, actually, a clone couldn't be an exact copy of yourself. It has to go through the same developmental process as anyone else.
Anyway, human cloning is very controversial. It's even illegal in several states.
Well, what's all the debate about?
Well, before we go there, let's make sure we all understand what we mean when we say ethics and policy. Ethics can be described as principles of conduct governing an individual or group. And policy, the way we use it in this program, means government actions or bills becoming laws. It's a debate about whether or not all cloning should be banned. There are two types of cloning: reproductive cloning, or cloning intended to create a whole new human being or animal, or therapeutic cloning, such as for research, with a technique called nuclear transfer. Reproductive cloning, or cloning intended to create a whole new human being, is illegal in some states. However, many states do permit therapeutic cloning, such as the cloning of human pancreatic cells to replace the damaged cells in a person with diabetes. This type of cloning may be a way to create new human embryonic stem cells that are genetically matched to a patient, avoiding any problems with the immune system. Although the ethics of cloning had come up before, the debate came to a peak in 1997 when Dolly was successfully cloned. This big advance in biotechnology raised a lot of important ethical issues and questions. Since then, some legislators in Congress have pushed for a ban on all human cloning. Although the cloning of cells may be very beneficial for the treatment of diseases, the possibility of cloning a whole new human being raises a lot of questions.
So the government really does make decisions about research.
Yes, and that's what this section happens to be about. Today we'll be talking about government policy, ethics, and science. In sections 2, 3, and 4, we covered ethical dilemmas about animal research, gene, and stem cell therapies. What this section will help you do is to put together the big picture. How do bills passed by the government affect scientific research? Where does ethics come in? And what may stop bills from going through? Probably a great place to start is to talk with Dr. Majumder, who is an ethicist at Baylor College of Medicine.
As an ethicist, my job is to address moral, legal, and public policy questions that come up in healthcare and in the biomedical field generally. Genetic research and biotechnology have generated extraordinary progress in terms of our ability to better understand and treat disease. At the same time, these advances have raised ethical issues that we as a society have never faced before. Much of the progress in the last 100 years is because of the work of scientists and their discoveries, everything from TV and computers to insulin and vaccines. So most of us would agree that science plays a very valuable role in our lives.
When you add all the diseases that could be impacted by stem cell research and genetic research, you will touch almost every person in this country, either with them or someone they love.
Although scientists have a lot of freedom to study what they want, the government at both the national and the state levels does play a role. For example, in funding research, sometimes that research money comes with strings attached. And on very rare occasions, the government may even tell scientists what they can and can't do. And that is true with some aspects of genetic research.
We've got to have solid, consistent funding in order to do the research. We need to be able to have the freedom and the resources to do what we need to do when we need to do it.
You have not only the tremendous potential to use stem cells for tissue regeneration and repair, there's a potential for abuse, there's a potential for fraud because of these strengths, and it really requires a lot of checks and balances.
Religious leaders ought to have a place at the table in the dialogue on issues of medical research, both in animals and in humans, because there's always going to be competing values. What the church strives to do in its moral teachings is to keep up with the medical sciences and to try to help them to understand that there is a moral dimension, an ethical dimension to what they do.
What the majority of scientists are interested in is not in creating a mini-me or an organ donor or even a winning football team, but in creating special kinds of human embryonic stem cells from cloned embryos. This is totally different than cloning a whole new human being. But still, some of the current genetics research can raise ethical issues. like the controversy about using human embryos for stem cell research.
I think the issues for the religious person begins with the question of where are these stem cells coming from, and are we getting stem cells by means of destroying a life?
From the Catholic perspective, life begins at conception. And so embryonic stem cell research literally destroys the embryo in order to use the cells as material for research to help other human persons. And so from the Catholic perspective, that would be considered immoral or inappropriate to destroy one human life in order to assist another human life.
The Jewish perspective, however, is very different. We don't see that human life begins with the fertilization of the egg cell. For the Jewish view, it doesn't become an independent human life until birth. Now, that does not mean that an embryo, a fetus inside the womb, doesn't have rights. It has rights, but only as a potential human life.
The main reason government plays a role in deciding how research should be conducted is that we, through the government, want to make sure that risks to people and to animals and to the environment are limited and that scientific activities are carried out ethically.
So if anything, the government is playing the diplomat, or in a stronger fashion, the police, to make sure people are following the necessary guidelines and proceeding cautiously with this science.
Government provides the funding and the appropriate regulations to make sure science is done ethically, particularly important in medicine, and also effectively. Part of the job of the President's Science Advisor is to advise him on where those decisions should be made and how they should be made so that science continues to progress.
Policymakers have a dominant role in scientific research because certainly for medical research funding, the lion's share of that in this country comes from the National Institutes of Health, which is governed by people who are appointed by the President and by Congress. And so they have a great say in what's funded.
The availability of funds at the National Institute of Health is really essential. The type of research that is being done is more expensive. The equipment that is needed is more and more sophisticated. Therefore, it's a critical period.
It's almost like embryonic stem cells have become more of a political topic than a science topic. And so I've done some writing, but a lot of the action on embryonic stem cells has been in Washington, where a lot of the policy debate about whether the federal government should fund research by scientists across the country.
So how does anything ever get decided on? And how does research move ahead if there's controversy about the funding to do it? First, let's take a look at the basic steps on how laws or policies are made.
The process of which a bill becomes a law is pretty simple, but it's a long process. It starts in either chamber, the House or the Senate, or they can start two processes together, and it would go through each chamber, the House. For example, if it started in the House, they'd have a committee hearing on the bill, and then it would go out of they take a vote, pass it out of committee, and it would go to the full House for a vote. Then it would go over to the Senate side. They'd also have a committee hearing, and then they would have a vote in committee and send it to the full Senate. Then it could go to the president. If it's a bill that started on each side simultaneously, or there's differences in the bill, it would have to go to a conference committee made of equal number of House members and senators, and then they'd reconcile the differences and then go back to each respective House and have a vote, and then go to the president, which he could either sign or veto. If the president signed the bill, then it would become law. If he vetoed the bill, then it would not pass, and it could go back to the House or Senate. Now, one thing they could do, though, is pass it with a 2/3 majority, which they call it veto-proof, so that where if it had a supermajority, then the president could not veto it. citizens and legislators can change the law. They can repeal it, or they can make amendments or changes once it's become law, and it's a very important process.
Ouch. Talk about what can happen to a bill. If you look at the history of government policy in human embryonic stem cell research, you can see what might happen when there are challenges involved. The President announced his decision today about allowing research only on a limited number of stem cell lines and prohibiting funding. Last week, Republicans and Democrats came together to propose federal funding go to research using human embryonic. Waiting for a vote in the Senate is a bill called the Stem Cell Research Enhancement Act of 2000. Religious cult claims to have produced the first cloned baby.
Talk about overload. We know from reading and watching the news that Congress has been introducing one bill after another about human embryonic stem cell research. In fact, there have been over 100 bills introduced in over 30 states either in support of or against human embryonic stem cell research. Why are there so many bills? What stops a bill from going through the process that we talked about? Let's hear about the debate concerning human embryonic stem cell research policies and what Congress is doing.
The challenge now is in those areas of medical research where, for example, embryonic stem cell research or therapeutic cloning, where people have understandable concerns, ethical concerns or religious concerns about this research. The challenge there is to get the balance right, is to make sure that what's being done is being done in a manner that is protective and sensitive to the special nature of human beings, because we all agree human beings are special. And right now we're in the middle of that policy debate.
Remember, the stage at which the embryo is in when it is used for research is smaller than a grain of sand. For now, scientists are still searching, and the debate continues. The stem cell, invisible to the human eye, may have some great promise in the treatment of diseases for the future. Yet, as you can see, there are issues that have stopped the progression of bills to fund human embryonic stem cell research.
Well, we talked earlier about how a bill becomes a law, but there are many factors in that process. One is public opinion. There's also lobbyists and organizations, key stakeholders that contact the legislators and let them know, maybe this isn't such a great idea. You started out with a good idea, but it's going to have these unintended consequences.
Healthy debate in Congress is a good thing. We don't want laws to be made without adequate consideration or without hearing different perspectives. I think the problem arises when the different sides don't listen to one another. That's when there's no movement on an issue.
Scientists always have tried to work together with policymakers. Not always their opinion goes hand to hand with the opinions of the government.
It is very important for science and religion, public policy, sociology, all of us, to work together.
When there's significant controversy over biomedical issues, we have some tools that we can turn to. In particular, there's a set of principles that we often use to help us analyze a difficult issue. Respect, non-maleficence, beneficence, and justice. These are a mouthful, but their definitions are pretty simple. In fact, you'll probably recognize these from personal experience. Ethical questions come up in our lives all the time, and we are always searching for ways to resolve these questions. Respect is acknowledging the value of each human being, letting people make decisions about their own lives when they are able, and looking after groups of people like children who aren't able to make their own decisions. Non-maleficence means avoiding harm to others. Beneficence is trying to help others, and justice is treating others fairly. Even when we use these principles, we don't always get an easy answer. That's especially true in our society, which is so diverse. The goal then is to better understand where we disagree and why, and to continue to search for areas where we can agree.
And so the dialogue becomes very important, science and religion, so that we can ensure that ethics, morals, faith, as well as scientific advancement are all able to be moved forward with the greatest amount of dignity.
The future of stem cell research is uncertain at this point, but my own guess is